In this lecture, we will cover the blood bank reagents that are used for testing within the blood bank department. In order to kind of help you comprehend blood bank as far as the reagents, you must first understand that blood bank is basically all antigen and antibody reactions. And in these cases, you have a known, which is your reagent, and you're looking for the unknown, which is going to be whatever is in your patient sample. So when we start off with our known, that is our reagents. So as far as our reagents go, we have known reagent antigens, so red cells. Here you have an image where you have some actual red cells where we actually know which antigens are on those red cells. So these are manufactured. Then we have antiseres. So anytime you see anti, that means it is an antibody. So an antisere is a known antibodies. So we know what is in these. So just like serum, sera is clear. So you get that clear reagent without any sort of cells in it. So our red blood cells, we have the antigens present on the red cells. So you're going to see red blood cells in that reagent. And then your antisera is going to be your antibodies. And so all that is in the sera is antibodies. So you can see here, this is anti-A. You can see back here, anti-D. So we know what is in these reagents. Then you have antiglobulin reagents. And so these are anti-IgG, or it's anti-immunoglobulin G. Or it could be potentially complement. And so we'll kind of talk about this in a little bit more detail. You can see an example of that here where we have the green bottle here that has the anti-human globulin in it. And then you also have potentiators that will enhance your antibody reactions. So we learned in immunology that certain antibodies, like IgG type antibodies, may have some weaker type of expressions. So sometimes in blood bank world, we need something to enhance that. So here we have an example of an enhancement type of reagent. Now, reagents do follow regulations, just like blood bank. So anytime there's any kind of manufacturing of any sort of blood product, whether it is a reagent or an actual unit of blood or platelets, fresh frozen plasma, anything like that requires that it go through an FDA process of regulation and approval. Now, so since it is something that is actually infused to the patient, it's kind of considered a drug. So that's why the reagents that are associated with that testing follows the FDA protocols as well. Now, the reagents must meet minimum standards in order to be considered to be used in blood bank. And so these standards state that it must meet certain specificity, meaning that it has to be specific for the particular antigen that you are looking for. And then it also has to have potency, so that you need to have a stronger type of reaction from that particular reagent. In other words, if it's an anti-D, that anti-D needs to be able to detect the D antigen at varying strengths. So at minimum, it needs to give what we call a 4+ reaction. Now we'll talk about these reactions in another lecture, but they do have standards that they must meet in order to be able to be used as a reagent. Now, just like any other reagent that's used in the laboratory, your blood bank reagents must be QC'd daily, and this is also going to include any of the equipment that you use, any kind of processes that you go through. There must be some kind of quality checks for those reagents and that equipment. Example of equipment check would be something like temperatures. So you would need to make sure that you are getting temps every day of all of your equipment. Other things that you might have to check, whether it's daily, weekly, monthly, whatever your schedule is for your lab, would be things like the speeds on your centrifuges to make sure that they are at the proper speed and that they're properly spinning your samples down to, or your test tubes when you're doing a tube blood type, to make sure that it's properly spinning that those samples down or those tubes down in order to get the intended reactions. And so there are various components that are included in a QC program, which again is going to include making sure that you have properly performed QC on those reagents every day. So each day, someone is responsible for actually testing the reagents to make sure that they're not denaturing over time with use, or they're not or haven't been contaminated or anything like that. So each day you're actually going to document those reactions for the QC of those reagents. So just like you would QC a reagent in any other area of the lab, you're going to test those reagents each day against known blood types to make sure that it reproduces the same type of reactions every day. Now, commercial antibody reagents can be made by a couple of different ways. They can be made to contain polyclonal antibodies or monoclonal antibodies. Now, polyclonal means that there's more than one. So these are made from several different clones of B cells that will actually secrete those antibodies of different specificities. And what will happen is there's different epitopes that will actually recognize those antibodies. An example of this would be anti-human globulin. Now, your monoclonal antibodies are, just as it said, single, so it's made from one single clone of those B cells that will actually secrete the same antibody with the same specificity. So it's only targeting one specific epitope. Now, it does use a hybridomal technology where it actually just recognizes that one single epitope instead of multiple epitopes for the same thing. Examples of this would be like anti-A or some of your other anti-serers, your anti-IgG antibodies. So just to kind of recap, monoclonal secreted by a single clone of an antibody, that antibody producing B cell. It's just one immunoglobulin class, either IgG or IgM, and it does have a unique specificity for just one particular epitope of an antigen. Your polyclonal, on the other hand, is secreted by different clones of an antibody producing B cell, so that means you could have a mixture of IgM and IgG antibodies. And you also have a mixture of antibodies that are directed against different epitopes of the same antigen. So for example, here at the bottom, we have monoclonal reagents for your anti-A and B. You can see it on the bottle that it's monoclonal. So that means that because it's monoclonal, it's going to have a unique specificity for one epitope of that particular antigen that you're looking for. So in this case, the A antigen or the B antigen. And then you can see where you have several reagents that say polyclonal. That means that they have specificity that is directed at different epitopes of the same antigen. So in these particular reagents, you can see being polyspecific means that it's going to target different epitopes that actually have IgG or, in this case, C3d. Anti-A and anti-B are the reagents that are used for determining your ABO blood type. And so this is the start of the reagents that gives us our forward grouping, or what we typically will just say what the patient's blood type is. So if we say they're type A or type B, we're getting this from the forward group typing. So this is where we use known anti-serers. So we use known anti-A and anti-B to type against the patient's red cells to look for a particular reaction. So if we have known anti-serer and we are trying to direct it against a patient's antigens, then those antigens are unknown. So if there is agglutination, that means that that antigen is present because we just put antibody against that antigen to see if it would agglutinate. So both anti-serers are directed towards a specific antigen that's on those red cells. So anti-A is directed against that A antigen, and anti-B is antibody B directed against the B antigens. And then we're looking for those reagents to have a formulation in which it gives those strong reactions, those 3+ and 4+ reactions, which we will discuss in another lecture of what those reactions actually are and mean. Now, testing is performed in an immediate spin phase, and that is because these are IgM type of antibodies. In order to confirm your reactions, then you would look to make sure that the patient does have the opposite antibody. So to do that, we would test the patient's plasma to see which antibodies are present. Additionally, when we're testing a patient's red cells, we are looking for the presence of not only your A and B antigens, but also a D antigen. So a D antigen is in the Rh blood system, and we'll discuss this in more detail later. But the D antigen is what specifies whether or not our patient is positive or negative. And so to use that serum or anti-D, what we're going to do is use that commercial serum. We're going to test against the patient's red cells or the donor red cells, and we're looking for that agglutination to happen. If that agglutination happens, then we know that that D antigen is present because we just tested it against anti-D or antibody D. If there is no agglutination, then that means that there is the absence of that D antigen. Now, additionally, one thing that we must do is to make sure that we have a negative reagent control to ensure that we do not get false positive reactions when we are testing our patients. Now, the makeup of D reagents or anti-D reagents can include high protein reagents or low protein reagents. Now, your high protein reagents contain polyclonal antibodies and approximately 20% of bovine albumin. Now, what this particular reagent makeup does is it actually promotes some false positive agglutination. So this is why we need that D control. Now, our low protein reagents, these are controls that ensure that typing results are correct. And so when we use these, we expect to not have any sort of agglutination. But if there is agglutination that is present, then it could be due to some sort of cold autoantibody or some sort of abnormal protein that's present. When is it required that we actually run this control? Well, it's required to run this control when you have RBC agglutination or red blood cell agglutination with all of your anti-sera. So in other words, your A, your anti-A, your anti-B, and your anti-D. So when you have agglutination with all of those, then you need to run a control. Because remember, we're testing patients' red cell for the presence of particular antigens. So if all of your red cells are agglutinating, then we're not really sure, is it agglutinating because it's reacting with the antibody that we just mixed it with, or is it agglutinating because there's something present in that patient sample that is causing some autoagglutination to happen. So a separate control is not needed if you have a negative reaction, because that means that when you've used those low protein reagents and you got that negative reaction, then you are capable of getting a negative reaction for that patient. It's only when you have all of those, all three of those tests positive that you need to be concerned that there is some sort of autoagglutination going on. So you'd run that auto control to verify. So just like we have commercial reagents where we have taken and bottled up known antibody, we also have reagents where we've taken and bottled up known antigens. And so these are actual red cell reagents in which there is an actual donor or person that has donated their red blood cells, and the red blood cells go through a testing process and a washing process. And then they're made into an actual reagent. And so we use these when we're testing for a patient's ABO blood type, as well as when we're looking for any sort of antibodies that could be present. So we would do an antibody screen, and if that is positive, then we would move on to do the antibody identification. So first we're going to discuss the A1 and B cells. So these are the reagent red cells that are used to do the reverse typing of a patient's blood type. So with our forward typing, that gives us our type. If it agglutinates with A, it's A. If it agglutinates with B, it's B. If it agglutinates with A and B, the patient is AB. If it does not agglutinate with either, then the patient lacks those antigens. That means they are type O. Well, the opposite is true for your reverse grouping. So you have to remember that we're actually testing patient's antibody which is the unknown here. So we're looking for these antibodies to be directed against known red cell antigens. So with our A1 cells, we have A1 cells present in that sample or in that reagent, and then our B has the B cells. So what this is an actual pool of donors or a single donor in which, they've been tested, and we know that they have just the A1 antigen or the B antigen on those cells. And when they're manufactured, they go through that process of washing and being resuspended to make either a somewhere between a 2 to 5% concentration, just depends on the particular reagent manufacturer. And then, of course, those patients or donors that are used to make these reagents, they have to be negative for Rh antigens because we can't have any kind of false reactivity. And they also something that you have to keep in mind is that when you're testing against these, the red cells are the known. Our patient's serum or plasma is the unknown. So when we test against the known cells, then we're going to get the actual opposite reaction. So that's why it's called the reverse grouping. So this particular test here, so for an A patient, then we know that they have the A antigen on their red cells, which means they should lack the A antibody, because this would be an example of them. It would be a patient that had, or this donor in this reagent cell would have A1 cells, or that A antigen on that cells. So if the patient is an A, type A, then they should lack that antibody, which means no agglutination. Now, one thing to keep in mind is that when you're using these reagents each day, you should visually inspect them to make sure that there's no agglutination in your vial, there's no signs of hemolysis, or that the cells look like they are the proper color, they don't have something growing on them. Now, as you can see here in this image at the bottom where I was just talking about making sure there's no hemolysis, these reagent red cells, this image actually come from the manufacturer's site, their website, but you can see here the supernate, this clear fluid here, and all of these tubes or vials is hemolyzed. But if you look at the bottom of the vial, you can see where the red cells have kind of settled out. Now, you can occasionally have like a little bit of a hue to the supernate, but it's never this bad. It should not be this bad. So if I were to be working in blood bank and I saw this, I would not use these reagents for testing, because obviously there's some hemolysis going on and it could potentially interfere with our reactions. But moving on. So our screening cells are used to screen for particular antibodies that may be present in a patient's serum or plasma. And so when you're doing an antibody screen, depending on the laboratory, they either use a 2 or a 3 vial system. So you can see here, this particular system is 2. You got your screening cells 1 and 2, and this one has got screening cells 1, 2, and 3. This ortho kit here, for their screening cells, they have 3 screening cells. And so each of these vials must be from a single donor or 2 donors that have been pulled together, and they should have a particular profile that we are looking for in order to give to have every particular or the particular antigens present on those cells. So when I talk about having particular antigens present on the cell, each lot of those screening cells comes with what we call an antigram. So it's going to look something like this here at the bottom. And so when we talk about the anagram and the cells that have to be present, that means that the FDA has regulated that this whole list here has to be present within that cell. So these donors, each of these is an individual donor or a pool of donors, but these single donors here have been antigen typed. So they've been antigen typed and we know which antigens are present. So each of these antigens, if it's present, that particular donor will have a plus, and if it's not present, then they will have a 0. Now we'll talk about these all in more detail in a later lecture, but for now just understand that each of these reagent cells, this would be a vial 1 and vial 2, are made from a donor cells, and they are manufactured in combo like this. So this set is set up together to try to have at least one of the donors be positive for these particular antigens that are present. So if you run through this list and look, you've got at least a positive and a negative present for almost all of these. There are some that don't have that particular antigen present, and we'll talk about why in a future lecture. Now the screening cells 1 and 2, or screening cells 1, 2, and 3, they do just as what their name suggests. They screen to see if there's an antibody present. If it is, then you would move on to the next phase of that testing, which would be panel cells. So the panel cells are very similar to the screening cells, except there are usually 10 or more vials within a panel cell. The panel cells are actually used to identify any sort of antibody that could be present, and each lot of those panel cells will have its own specific antigram profile with it. So you just see kind of an extended version of what we just looked at. So in our last picture, we saw where it was just a 1 and 2 donor. This particular panel here has 11 donors, and each of these individual donors has their antigen profile makeup listed on this panel. So we can see which ones are positive for the D antigen versus which ones are negative for that D antigen. So you can see your negatives here versus your ones that are positive. They're here. Now, we'll talk about how to actually interpret and work this up a little bit later on, but it's going to be based off of the particular reactions that patient reacts with those particular reagent cells. An antiglobulin test. So the principle of this test is that you have some sort of commercial antibody with a specificity towards human globulins, which is used to agglutinate any sort of antibody-coated red blood cells. So you can see here in this image where you might have an antibody-coated red cell. So you have all these antibodies that are attached to this red cell here, and then you come in behind that and you plug in some anti-IgG or anti-human globulin that will actually attach to those IgG antibodies that are present there. So we don't really necessarily know which particular IgG antibody, we just know that there's one attached. So we're looking to see if there's something attached. So reagents that contain antibody towards these particular antibodies are going to include your IgG or anti-IgG, as well as complement, so anti-C3d or anti-C3b. There are two types of antiglobulin tests, direct and indirect. With your direct antiglobulin test, it detects IgG or complement that is actually bound to the cells in vivo. So when we talk about something being in vivo, you got that V in there. That means it was in the vein or in the patient. So we're looking to see, has any sort of IgG or complement actually been bound to the patient's red cells while it was in the patient's body? So in order to do that, we need to figure out, we need to first take that anti-IgG reagent and mix it with that patient red cells to see if we get that agglutination where those cells have already been coated with IgG. And in this procedure, you have to actually wash your red blood cells before you add that reagent in order to remove any sort of unbound proteins. And then agglutination demonstrates whether or not there's any sort of IgG or complement once you actually get that, you read that reaction of that test and you see that there's agglutination that's happening. Now, some examples of when you could actually get positive reactions would include things like transfusion reactions, hemolytic disease of the newborn, autoimmune hemolytic anemia, and drug-related mechanisms in which there's some kind of IgG drug complex that takes place. The other antiglobulin test is the indirect antiglobulin test. And so what this does is it actually detects IgG or complement that is bound to the red blood cells in vitro. So in vitro, you got that T in there. That means in your test tube. So this basic step is your antibody screen, or your antibody ID. So we are looking for some sort of indirect coding from those antibodies on those red blood cells. So if a physician calls down and requests an direct antiglobulin test, or well, actually they'll call and say, I need you to do a direct and an indirect antiglobulin test. Well, typically you've already done a type and screen, so that antibody screen is in that type and screen. So you've already actually done that indirect antiglobulin test. So if you ever hear anybody referring to an indirect antiglobulin test, it's just basically your antibody screen. Now, additionally, the cross-match and antigen typing is part of an indirect antiglobulin test, and that's because you do take them all the way through that antiglobulin phase of testing. Now, there are some situations where you can get false positives with this testing, and that would include when you have some red blood cells that have agglutinated before you actually did that wash step, and then you add that anti-human globulin. And so this could happen from a false reaction for something like a cold agglutinin or cold reactive antibody that's caused those cells to already agglutinate before you even go through that testing process. Using dirty glassware, so if you got particles or contaminants in that test tube that's causing that false reaction. And of course, improper centrifugation or centrifuging too long can cause that red blood cell to pack really tightly, and then you're unable to kind of shake that off of the bottom of that glass tube, and then you get that non-specific clumping that's going on that's really just kind of false. Now, there are several things that can cause false negative antiglobulin testing. And now when I say antiglobulin testing, I'm referring to direct and indirect testing. So false negatives can include failure to properly wash those cells, if there's a delay in your testing when any of those steps are coming about, failure to actually notice and recognize those very weak reactions. So you can actually shake a tube too hard and break up those reactions. If your reagent loses its activity, so it just kind of denatures over time, either how it was stored or if it's contaminated. A failure to actually add that AHG reagent. This is one of the reasons why a lot of people opt to use the green reagent, so you can actually see it. If you use the clear reagent, you can't really, sometimes it's a little bit harder to see it in that tube, so a lot of people like to use that green reagent. Not properly centrifuging it, so you didn't centrifuge it long enough. Using an inappropriate suspension, so you make your suspension too heavy or too light, so you're not within that 2 to 5% suspension that you make, and we'll discuss how to actually make those suspensions later. Your antiglobulin reagents can be polyspecific or monospecific. So with poly, they're going to contain both IgG and C3 antibodies. They are derived from a polyclonal or a monoclonal source. If there's agglutination, there indicates that there is IgG or complement that's coating those red cells. If positive, then you would actually need to determine, is it IgG or is it C3? So here in this example here at the bottom, this middle reagent here, you can see that polyspecific, meaning it has the IgG and C3 present. And then you have the monospecific. So don't confuse this with clonal. It's a little different. So clonal is how it's made, and then the specific means which ones are actually in there. So polyspecific, polymultiple, and then monospecific. So you just have the one particular antibody that it's targeted against. So you can see these reagents here that is just IgG present. And then over here you have this reagent that has just the C3 present. Now your C3, C3b, C3d is actually looking to see if you've got the activation of that classic pathway, looking for intravascular or extravascular hemolysis in that patient. Now, as we discussed, there's many false negatives that can happen with your antiglobulin testing. So when this test was originally kind of discovered, made, it was done by a man called Coombs. So you actually will often hear Some physicians still refer to a DAT as a Coombs test. And so Coombs is not really a test, it's actually a dead man. So Coombs refers to the man that actually developed this test methodology. And so you actually sometimes refer to check cells or Coombs control cells. But what I'm trying to get at with this is we saw where there's a lot of false negatives that can happen with that particular test method. So a way to ensure that you kind of catch those errors, there are cells that are made that are already pre-coded with that IgG. And so when you drop that into your tube where you already have your patient's sample in it and you've added that AHG reagent, then if your patient was false negative, in other words, that they really should agglutinated, then when you drop this reagent in there, it's not going to agglutinate. So you need these cells to check to make sure that it really did agglutinate, that you didn't get a false negative. So if you have a negative reaction and you add that reagent or these check cells into that tube and you get that reaction, that positive reaction, then that means that that reagent, that whole process worked. So it's not anything that you did, it's that patient was negative. So you always use check cells for those reagents. But if you hear someone refer to a Coombs test, they're just talking about that DAT test because the test was named after the inventor or whoever come up with that test. Now, when we start learning about the different antigens and antibodies that a patient could potentially have, then we're going to kind of start learning that they each have a different mode of operation. So, and that has to do with their reactivity, how they respond in a sample, how they interact, those antibodies interact with those antigens when they're presented in there. And so some of the things that we can do is kind of enhance that reactivity to speed up those reactions. So we can do that by adding potentiators. And the potentiators, there are a few different ones. So depending on your lab will depend on what they use. There are things called low ionic strength solution. Or low ionic strength saline. What this does is it increases the rate of antibody uptake. You also have PEG, and what this does is it concentrates the antibody in the test environment in a low ionic strength saline. You also have other proteolytic enzymes that can be used to alter the charges of the red blood cell membrane, which will help reduce that zeta potential. In some of the particular red cell antigens that are present, it can actually denature them. And then one of the additional ones that can be used will be bovine serum albumin. And what this does is it reduces the repulsion that happens in between cells. And so it helps to reduce that incubation time that we typically will use for the particular test method. Now, bovine serum albumin can actually come in different concentrations, so that will just depend on what your lab's preference is as far as what they will use. And as I previously mentioned, it does allow those red blood cells, once they've been sensitized and they've been coated with those antibodies, it allows them to draw a little bit closer together. And it favors direct agglutination with Rh antibodies, so it helps when you're trying to enhance any potential Rh antibodies that could be present, and it enhances the sensitivity of that indirect antiglobulin testing. So when we're doing that antibody screen, antibody ID, any sort of cross-match or antigen typing. Lectins are seed extracts that have a specificity towards a certain red blood cell antigen. And then what they do is actually bind to the carbohydrate determinants of those particular red blood cell antigens. And so these reagents are actually used looking for more or less when you're troubleshooting certain patients. And so we'll learn a little bit more about how these are used later on in later lectures. Now in blood bank, you can do tube testing. Another alternative to tube testing would be using a microplate. And what that does is it just kind of reduces the volume that is used, and it kind of just simplifies it down into one big plate for all of your reactions. These look very similar to the microtiter plates that are used in other areas of the lab, like immunology, micro, etc. And so it applies the same principles as your tube method, where you're looking for that agglutination of that solid button in the bottom of that well, versus where it just kind of disperses out and moves around where there's no agglutination that's happening. We will talk about this in a little bit more detail in another lecture. Gel technology. So what this actually does is it uses a gel particle or little tiny gel beads to trap any sort of agglutination or aggregates of red blood cells. Now inside of these wells, so you can see your gel wells here, or your gel card, and then you have those wells within that. So inside that well, you have all of these beads of little gel particles that are kind of bumped up against each other. Now above it in this little area here, you mix your patient sample or your reagent red cells or whatever goes inside of this, and then it goes through an incubation step or a centrifuge step or both. And then when it's in that little incubation period, you're allowing that reaction to take place between those red cells and those antibodies. And when that reaction takes place and you start getting these aggregates of cells that have agglutinated together, then at first these red blood cells are smaller than these little gel particles. So when you spin it, they just kind of all start kind of moving through individually to the bottom. However, if you have these larger clumps, then what happens is they get hung up and they're not able to pass through that gel matrix, and so then they sit in the place wherever they're at. So you can see the different types of reactions. So if you have agglutination and those cells are unable to pass through and you have that solid band across the top, then you have 4+. If it goes from the top to about midway, that is 3+. If it's from the bottom all the way through, or pretty much from the bottom halfway up, that's 2+. And then if it's concentrated just to the lower part of that well, it's 1+. And then you have all your cells go to the bottom is negative. Next we have solid phase. So solid phase uses those microplates as well. The only difference here is that the reagent is actually inside of, or coated inside of those wells. So what will happen is if there is a positive reaction that takes place, then that antigen-antibody reaction actually takes place on the sides and the bottom of those wells. So for a positive reaction, you will actually see where the whole bottom of that well is coated. A negative reaction, when you spin this down, or when it sets, you actually have all the cells will just kind of start to flow down the sides of those wells and kind of migrate down till they form one little button in the bottom of that well. So this would be considered a negative reaction, and then these are all positive reactions. And so it's kind of the opposite of what we look at in tube, because this does look like, it looks like a little agglutination, like a button. However, it's not. It's just the fact that all of those cells have kind of settled down into the bottom of that well, whereas these cells have all attached to the side of that well. So let's just say your well looks like this. A negative means they've all kind of come down here to the bottom. A positive means they don't really ever make their way down there, because they're just kind of sitting and hanging out along the edges of that well. And this concludes our lecture.